Hello, I am Dr. Jose Saavedra. I'm a pediatrician, a gastroenterologist at Johns Hopkins University, and it is a pleasure for me to join you this evening to go over some concepts that we hope will enhance the way that we manage and help our kids in this world, in these hard times, with the goal of ultimately preserving health for a long time. My thanks to the Nestlé Nutrition Institute for this opportunity to reach colleagues half around the world, and my hope is that in the end you'll have something positive to take back with you to your daily practice. We have come a long way in understanding what and how growth and health should be managed for the first few years of life, and a tremendous amount in the area of pediatrics has been learned through the understanding of what and how human milk and breastfeeding can help support health of children. Over the last 50 years, we've had a tremendous amount of improvement in the number of infectious diseases. Today's COVID-19 epidemic actually shows us how quickly, despite the tremendous challenge and cost that this infection is going to cost us, how quickly we can overcome infectious diseases. In fact, over the last 5 decades alone, diseases like rheumatic fever, which is a streptococcal disease, or tuberculosis, mycobacterial disease, or other viral diseases like mumps, have been dramatically reduced in the world with all the work that has been done from the healthcare point of view. That said, in the same period of time, we have seen a tremendous increase in a number of non-communicable, mainly inflammatory conditions, like Crohn's disease, type 1 diabetes, allergic asthma, that have mirrored in the opposite direction what has happened with infectious diseases. And together with these, and compounding the problem, has been the increase in the changes that relate to the body mass index and the adiposity of the population, and particularly children, which have led to an epidemic of increased conditions associated with obesity, which starts in childhood. Now, from the nutritional point of view, similarly, on the left, we see a significant decrease in what has been malnutrition or chronic malnutrition, such as stunting, while at the same time, we see this dramatic increase in the frequency of obesity in the pediatric population worldwide. The number of obese children under 5 years of age today continues to grow and is reaching epidemic proportions. Now, these non-communicable diseases are today responsible for more than 40 million deaths each year. And that is more than 71% of all deaths globally. In India alone, close to 6 million people on a yearly basis die of non-communicable diseases rather than infection or malnutrition. And childhood overweight and obesity, in particular, are responsible for the epidemic that we have in older individuals that relate to diabetes, hypertension, and cardiovascular disease. What we're going to review today is just 3 of the key areas where some of our understanding on human milk and breastfeeding has led to the knowledge that helps us modulate how metabolic responses that may be related to overweight and obesity, how neurodevelopment can be modulated with nutritional interventions, and ultimately how our immune response, all of these activities which ultimately set the course for long-term health. And as we said before, in pediatrics, we have this great advantage that breast milk gives us this evolutionary model that inspires how we should feed children. Children who breastfeed have a lower risk for overweight and obesity. On the left, children who are never breastfed will always have a higher risk than children who have any breastfeeding as a population. And not only that, the duration of breastfeeding, as we see on the right, can decrease that risk significantly. Now, children who are not breastfed obviously are receiving milk from other mammals. Cow milk is very different from breast milk. It was meant to feed calves, not human babies. The protein content in particular in cow milk is very high. This has led to a number of speculations and now better defined hypotheses such as what we call the protein hypothesis, which suggests that infants who do not get breast milk and get milk from other mammals, particularly cows, will increase the amount of protein they get, increase the amount of insulin generation amino acids, which can lead to weight gain and adiposity, and ultimately risk of obesity. High protein in general has not been supportive of adequate growth. When we look at in orange, the breast milk content of protein, it evolves over time. And historically, we have been giving children significantly more proteins in milk diets and in infant formulas. Worse on when they get cow milk. Now, these high levels in infant feeding of protein were used because the quality of cow milk is not the same as that of breast milk. Therefore, we always wanted to give higher levels of protein for the purposes of justifying and providing the adequate amino acids that kids need to grow. Today, newer technologies that can modulate the quality of protein used in milk diets of infants can significantly improve the amount of essential amino acids, can lower the total protein that we give children, so we can give high quality in lower amounts, which is more in line with what the nutritional requirements of infants and children are. And there is increasing data to demonstrate that lower protein intake can actually decrease the risk of overweight and obesity, at least all the way to 6 years, and in epidemiologic studies for a longer time. Children receiving lower protein infant diets in the first 2 years of life can decrease their risk of obesity by about 50%. That is about a 5 to 6% reduction of obesity in the population of otherwise healthy children. Now, when we talk about protein quality, and again, we need to understand a little bit of those drivers of what modulates the quality of the protein that we can provide children. Milk of other mammals, and particularly cows, are predominant in casein. Casein is that curd-like component that happens with acid digestion. Casein is adequate for other digestive systems. Less so for the human, because the large amount of curd will also lead to other complications that we'll mention briefly. Human milk, on the other hand, is 70% soluble whey protein, less casein protein component. And this alone can modulate some of the digestive processes that occur in children when they're getting milks that are lower in casein curd components. Whey protein alone, when we compare whey protein to casein or to soy proteins, whey proteins have a significant higher protein quality when we measure it in the as total amount of essential amino acids, or protein utilization, which is the amount of nitrogen used versus that is ingested, or protein efficiency, and that is the weight gain per gram of lean body mass per gram of protein that is ingested. In all these measures of quality protein from WHO and FAO, whey is a superior protein in terms of delivering a better nutritional alternative. This higher component will allow us to give better protein with less curd formation, which also helps decrease the amount of time that milk stays in the stomach of children, and that is an improved gastric emptying with higher whey protein. Switching to what and how we have learned in terms of neurodevelopment associated to human milk and breastfeeding. The vast majority of mechanisms that mark the development of the human brain and its activity, whether it's just hearing and sight, cortical functions, receptor and language functions, and ultimately synaptogenesis, which is the connection of all the neurons, between the neurons that have to happen in the brain, all happen in early life. And it is very clear that in the first 2 years of life, the brain growth is at its highest peak of acceleration, and a number of factors play a role. Clearly, The green line, which is the high socioeconomic strata rate of gain of gray matter of children, is much higher compared to children in low socioeconomic strata. And this may be due not only to a number of nutritional and growth factors, but to the environmental stimulus that these children may not be getting. And when we look at the more direct effect of nutrition, clearly children who are stunted, and that's the light blue bars, will have a lower amount of gray matter over time than children who are well nourished. Nutrition in these 2 years plays a tremendous role. And we can see that in function. The general cognitive ability, language ability, and the motor skills of infants in early neurodevelopment is also improved in children who are breastfed, that's the blue line, versus children who are not, and that's the red line. Now, these factors of neurodevelopment also can be broken down into a number of different components. Neurogenesis, the generation of neurons, obviously begins in fetal life and continues in the first few months. The second component, which what we call myelination, is probably the most prominent from birth to the first year of life. And myelination is the generation of the myelin wraps of tissue, most of which is fat tissue around the axons of the neurons to help them connect faster from one neuron to another. This will lead to more neurons synapsing together, and creating dendrites and synapses, and of course we need synthesis of neurotransmitter over all the period of life for adequate information to go from one neuron to another. These different stages or components of neurodevelopment are all related to the specific nutrients found in human milk. In particular, myelination seems to be one that takes up a very large amount of energy and a number of nutrient factors, including long-chain unsaturated fatty acids like DHA, phospholipids, choline, iron, and iodine. All these are critical sensitive nutrients for the development of adequate myelination, which ultimately leads to better neuronal communication in the brain of infants. One quick word about each one of those. DHA in particular, and we've learned this over the last 25 to 30 years. Long-chain unsaturated fatty acids and DHA specifically are critical in facilitating the development of myelin tissue and in general brain growth. Interestingly, the amount of DHA in human milk of a mother highly depends on her diet. And when we look at the levels of DHA in human milk, these vary by 10 to 20-fold, depending on the population that we look at. The median concentration of DHA should be roughly about 0.32 milligrams of percent of total fat. And that is a median. That means there's populations that are significantly higher and significantly lower in DHA. Those populations that live in coastal areas and consume large amounts of shellfish and fish will have higher levels of DHA than those that don't. In India in particular, the rates of consumption of DHA are significantly below what is considered the RDA of 300 milligrams for mothers who are lactating. Phospholipids are another whole area of understanding, and the presence of high fat content and particularly phospholipids and choline facilitate the creation of sphingomyelin and phosphatidylcholine, which are the two most important components of the myelin sheath of fat, which is about 70 to 80% fat. These ultimately are critical so that this fat wrapper of myelin in around the axons of neurons can facilitate the speed with which neural signals travel. And ultimately choline, which is a precursor of many metabolites that ultimately will form the structure of the white matter of the brain, together with fat and combining DHA with choline will support the creation or the generation of phosphatidylcholine much more efficiently than choline alone or DHA alone. And the combination, again, from the understanding of the composition of human milk, of these two components in the diet, clearly sets the stage for an adequate delivery of these nutrients over time. And it's clear now, and this is work just over the last couple of years where we have finally begun mapping the evolution of white matter in infants and children. As you see from this graph, children receiving exclusive breast milk in the first 3 months of life will have a higher, and this is across the board and statistically significant, a higher level of achieved total white matter in multiple areas of the brain when we compare it to children who are receiving other milk diets. Finally, let's briefly look at one final aspect that relates to our learnings from human milk and breastfeeding compared to children who do not. The immune system obviously gets shaped and modulated and adapted in the first months of life, and we are aware from multiple epidemiologic studies that breastfeeding can protect both from infections, respiratory, gastrointestinal, and others, as well as from immune-related diseases in children. And we're just beginning to understand the mechanisms by which this can happen. One of them, and back to the importance of fat in the diet of children, as much as we have learned from the content of fat, and particularly DHA and ARA in breastfed babies. These fatty acids also not only modulate brain function achievement, and both structurally and functionally, but also modulate neutrophil function, neutrophil activity, phagocytic activity, all of which modulates also the transformation or the changes that occur from CD4 cells to T helper cells, which have led to decreased or greater evidence for the protection from bronchitis and bronchiolitis, from other respiratory conditions, from allergic diseases, as well as from a number of inflammatory processes that can be minimized by the adequate nutrition of children, again, using human milk as an example. And very importantly, it is human milk and environmental factors which also lead to the modulation and the generation of the correct amount and the correct type of microbiota in children. Children are born with a relatively naive adaptive immune system, but it is the inclusion of bacteria in the GI tract that ultimately charges the immune system, trains the immune system, the gut-associated lymphoid tissue, where the majority of our immune cells lie, to ultimately have a well-adapted immunologic response. The presence of bacteria in the gut is the major determinant of the immune development and the immune response of a child. And the balancing of this microbiota with a predominance of specific non-pathogenic bacteria, like bifidobacteria, seems to be the key to an adequate, balanced immunologic response. There are many factors that affect the microbiota composition of infants, including a very strongly very strong impact of how a child is born with a vaginal delivery versus a sterile C-section, and receiving human milk, which has not only bacteria, as opposed to milk diets that are pasteurized and contain no bacteria or probiotics, which promote this balanced microbiota with predominance of bifidobacteria, while C-section feeding sterile foods, including sterile milks, the use of antibiotics, and of course the use of environments that minimize the inclusion of bacteria all promote what we call dysbiosis. And dysbiosis is this imbalance of beneficial bacteria, which leads to lower gut barrier function and inadequate immune response. And a number of conditions, infectious, allergic, including necrotizing enterocolitis, are associated with this dysbiosis. And it's been really more than close to 75 years where people discovered or began describing that human milk has a factor that promotes the growth of bifidobacteria. And if fast forward several decades, it is very clear that a number of these factors in human milk play a role, including the fact that there's a lower protein and a lower phosphate content in human milk. In fact, that lactose is the predominant sugar, which also promotes bifidobacteria. And more recently, we have now describing what it is that the components, or what components, particularly carbohydrate components of human milk, can promote growth of specific species like bifidobacteria, which has led to this concept of human milk oligosaccharides. Human milk oligosaccharides, or HMOs, are very complex molecules of glucose, galactose, galactosamine, or N-acetylglucosamine, fucose, and sialic acid. We don't have time to review the whole structural understanding that we have of HMOs, but it is important to understand that when we look at just the solids of milk, HMOs are one of the largest solid components of milk in humans, which we have just began describing. There's close to 200 different structures of oligosaccharides, or HMOs, in human milk, of which 2-fucosyl-lactose, the one with the dark fraction of the pie there, is the most abundant. And a number of other oligosaccharides, including another one that has recently been synthesized, lacto and neotetrose, or LNNT, conform close to 10% of these oligosaccharides in human milk. We don't have time to discuss all the effects, but it is very clear these are very active molecules which directly and indirectly modulate, first, the intestinal microbiota with this preponderance of bifidobacteria. But they also have direct effects by deflecting pathogens and serving as decoys for pathogens, helping to strengthen the structure of the GI tract, and ultimately educating the immune system. This very complex understanding has been really only in the last 2 decades, but it is very clear that these HMOs have ultimately also a clinical effect. When we look at mothers with high levels of 2-fucosyl-lactose, which we mentioned is one of the largest components of HMOs, women with high content of 2-fucosyl-lactose in their breast milk will all have reduced frequency of infections, in this case, respiratory infections in early life. We don't have time to discuss a number of studies and literature which is increasing, documenting that 2FL in human milk is associated with a lower incidence of a number of infectious diseases, in this particular case, diarrhea, not only in frequency, but also in morbidity. So in this case, biology in mothers with higher levels of 2FL is showing us that there is a clinical effect of these molecules. And only recently we have begun synthesizing these so that we can actually do prospective randomized studies. And for example, infants who received diets with higher levels of 2FL and LNNT, 2 of the main oligosaccharides in human milk, report fewer episodes of bronchitis, fewer episodes of lower respiratory tract infections, and lower use of antipyretics and antibiotics in the first year of life. We have yet a lot to learn, but it is very clear that these molecules have a significantly higher activity than what we understood until recently. And it is important to note that HMOs that you see on the right, and these 2 examples, 2-fucosyl-lactose and lacto and neotetrose, are very different than other oligosaccharides, which we also have learned a lot about. Galactoligosaccharides and fructooligosaccharides, for example, are 2 of the most commonly used oligosaccharides for infants, which are very different than what is in human milk. They do have a predilection for being fermented by many species in the microbiota, but HMOs have a high selectivity for being fermented by bifidobacteria. GOS and FOS are not present per se in human milk in any amounts. Fructose, which is part of FOS, is not present, of course, in human milk, and they don't have all the other complex molecules like fucose or glucosamine. Therefore, they are going to be functionally very different from human milk oligosaccharides. And finally, there's a number of studies that are just beginning to emerge showing, for example, that the use of galactoligosaccharides compared to breastfeeding or compared to the use of HMOs, in this case 2FL at different concentrations, GAUs will actually increase the levels of pro-inflammatory cytokines because a large amount of fermentation is occurring by a number of species versus a decrease in inflammatory cytokines associated with breastfeeding or with adding HMOs to infant diets. So to summarize, breastfeeding, and with these examples, I hope I've shown that it remains the model for nutrition in early life and ultimately sets this course for long-term health. Breast milk is whey predominant with higher amount of protein quality with a lower amount of total protein, which may help adequate growth, metabolic programming, and decrease the risk of obesity. DHA, ARA, phospholipids, and choline remain one of the key nutrients for adequate brain development and cognition of infants. And HMOs, which we're finally understanding, can significantly impact the development of a healthy immune system in infants and children. So I will pause at this point and take a few questions, and I think we're receiving a few now that I can see on the screen. For vegan mothers and lactating mothers, would you advise on a DHA diet-rich diet so that babies don't reach DHA stores? The answer is yes. Vegan mothers in particular, right? If they are not going to be able to consume shellfish and fish, particularly lower density fat fish or fish with fat, most likely will be too low on their consumption of DHA. And the current RDA of 300 milligrams can easily be done as a supplement. Another question, in the absence of breast milk, how do we ensure lower delivery of protein similar to human milk? Today, for infants, from the nutritional point of view, it really is only well-studied and approved infant formulas that can or should be used in the absence of exclusive breastfeeding, either to complement human milk or to replace it if a child, for some reason, unfortunately, cannot receive those benefits from exclusive breastfeeding. And today, as I mentioned before, newer infant formulations for infant diets have been developed that decrease the amount that we historically gave of 2 grams per 100 calories, and when we use raw cow milk or just pasteurized cow milk, 3 to 4 grams of protein per 100 calories, which is far in excess of what the infant needs. Adequate infant formulas and diets with lower protein content that are now better developed are the only real alternate choice for doing that. Another question is on the benefits of human milk. So what are benefits of human milk oligosaccharides? So I think we've mentioned a few already. And let me just highlight the fact that we understood now for some time that providing non-absorbable sugars, oligosaccharides that are not lactose or only glucose or fructose, which we really should not be feeding infants, those sugars, when not absorbed, are fermented by bacteria. These bacteria grow and, of course, per se, will modulate the immunologic response of the infant. The advantage of human milk oligosaccharides compared to other non-absorbable sugars like GOS and phos, for example, is that not only they are selectively fermented by certain species, particularly bacteria, but also seem to have very direct effects that are not obtained with galactose oligosaccharides or fructooligosaccharides for that matter. Fructose does not exist in human milk. These oligosaccharides will be fermented by a variety of species. HMOs will have preference and be more selectively fermented by very specific species. as I said, bifidobacteria being one of them. But they also serve as decoys. HMOs can act as sugars blocking the attachment of pathogenic bacteria onto the gut epithelium. This is now, we know from animal studies, and as I mentioned, the clinical data is just beginning to appear. Another question is on breast milk. Again, I hope we've answered some of it. How does breast milk help cognitive development of the infant? I think I've mentioned a few. No doubt part of it has to do with the nutrient composition of human milk. It is important to understand that the majority of brain tissue is actually fatty tissue. And it's this very complex composition of fat polar molecules, particularly phospholipids in both gray matter, but particularly in white matter, that seem to play a huge role in what ultimately is development of cognitive function. Today, I think the understanding of the role of DHA and ARA and phospholipids and choline in human milk has also led to modifications in the diets of children who, for one or other reason, are not getting exclusive human milk, so that we can hopefully minimize this disadvantage that non-exclusively breastfed babies get when they don't get only their mom's milk. Looking for any other question. One other question relates to the better understanding of what and how human milk ultimately has led to the adequate adaptation from the evolutionary point of view. It's very clear today that no food is perfect, obviously, but the beauty of lactation is that it has taken hundreds of thousands of years for this type of exclusive feeding with one food, human milk, can help increase the chances for survival. And in the first few months, that has not moved the needle at all in terms of understanding that exclusive breastfeeding in the first, especially in the first 4 months of life, but through the full first year of life, brings the child a number of benefits that have been very difficult to mimic. We have made tremendous progress in the understanding of what a child needs to grow. We're now better understanding of what a child needs to function appropriately, and not only to have adequate weight and height, but also to modulate the different components of lean body mass and fat tissue and immunologic response and brain function. All these we have gradually learned, and again, human milk continues to be the best way to measure and understand how we can get those benefits. So, I don't see any other additional questions coming in. I really hope this was helpful to you all. I wish everybody to stay safe, to do our best in communicating, again, using technological means while we have to minimize the amount of contact that we have. But hopefully we'll overcome this new area of, or new era of COVID-19 and ultimately lead ourselves and others to continued health. So I wish you all a very good evening. Thank you very much.